Hello everyone, welcome to our channel Applied Forensic Research in Sciences. My name is Diksha Mehra and today I'm here with the first practical experiment session from AFRS. So experiment number 1 is based on Cheiloscopy, that is the study of lip prints. So to begin with the experiment quickly, our aim is to develop and identify lip prints. For today's experiment, we'll be developing and identifying my lips. So these are the sample lips. And to begin with the experiment now, I would like to introduce the material we'll be working with. So this is a soft Bristol brush, fluorescent powder, gloves for our protection and the integrity of the evidence. Also a mask, because these fluorescent powder we work with are carcinogenic in nature. So to avoid them entering our nose, nasal passage or even our mouth, so we need to take precautions. So it's important to wear a mask. Blank paper and the sample lips which are already here. So now to begin with, I would first tell you a little bit about how we take lip prints and why we take lip prints basically. So in 1902, there was a renowned anthropologist named R. Fisher. So he was the first scientist who actually discovered the fact that there are some furrows present in our lips. Like, this is something very common. We often day to day, sometimes many times in a day, we look at our face in a mirror. But do we ever notice that there are some specific patterns which are present over lips? So there are two major parts in our lips which tell us about the pattern. This outer lining is called the vermilion border. This border. And the area inside is called the vermilion zone. On this vermilion zone, there are patterns present both on the upper lip and on the downward lips. So now we are going to develop pattern present on the vermilion zone. Now, in 1932, Edmund Locard was the first scientist to use these lip prints for personal identification. So basically, within this span of 30 years, they were able to study that these patterns present are actually unique. So they can be used as a potential biometrics for an individual. So now we'll quickly begin with the procedure of this experiment. As we discussed that the principle is necessarily the uniqueness of these patterns which can be used as a potential biometrics for an individual. So, please wear your gloves. It's very important both for our own protection and for the integrity of the evidence. So, don't touch the brush or the fluorescent powder or anybody's lips like this. So, make sure you are wearing gloves. And I won't be putting on a mask because I'm taking sample of my own, and the fluorescent powder that I am using right now is safe to use, even though it will still be a little harmful for the nasal passage, but I would be very careful for that. So this is my paper. It's a rectangle sheet. Now what I'm going to do is horizontally I'll fold it in half. And the sample which I'm going to take would be on the folded line. When you are taking sample by yourself or you are taking sample of somebody else, you need to ask them to relax. My lips are well moisturized right now. When you are taking somebody's sample, make sure that their lips are not too dry, not overly moisturized, because then they will smudge the patterns. And if they will be too dry, the pattern won't be able to come properly. So if you feel they are too dry, make them use a lip balm or any other glycerol or a moisturizing gel to soothe them a little. And if they are overly moisturized, so ask them to damp it with a tissue or something. So now they are in a good condition for me. Now, you have to place this paper from the both side on the edge of the vermilion border, that is this, till here, on both the directions it has to go. And once you place it there, ask them to slowly put their lips down on the paper, but pattern them nicely on the paper. That is, press them nicely, not too hard, not very gentle, but they have to place it properly. When you will be taking this paper out, ask them to slowly open their mouth and do not make any swift movements to pull this paper out. It will smudge the pattern completely. So then gently you have to take the paper out. So now I'm going to demonstrate it. So I've got my pattern over it. It's slightly visible in the light that yes, there is something. Now, another important note over here, this pattern is wet right now. Even if you feel like no, it's not wet, it's dry, it is wet. So you need to leave it in a shady place. Do not make it sun dry. It would harm the particles of saliva in case they are present. Moreover, do not use any external heating source like a hair dryer or a another kind of electronic device for drying or heating the paper. It might damage your pattern as well as some other tiny uniqueness of the lips present. So avoid using any heat source. Now, by the time the pattern is drying, what we can discuss is that where are these lip prints being used? See, lip prints are a necessary biometrics, as I told you. But then we all will be like, we already have our fingerprints for biometrics, which work very nicely. So, how lip prints can help us? So, basically lip prints are important in some specific cases, yet they have a very significant role to play. Likewise, for cases having sexual assault or cases of kidnapping or even cases in which poisons are involved. Likewise, much of our poison traces are left on our lips, especially when they are given in powder form. Moreover, whenever we are drinking something from a glass, our lip prints are always present on the glass, no matter what nature the glass is of, even if it's made up of the glass material, thermocol, paper glasses, steel glass. We can always develop lip prints from these sources also and give us response if we should make a video on different sources from which lip prints can be taken on, if they are present on different materials like steel, glass, paper, like another paper. Even on spoon we can have potential lip prints, because lip prints are not much even the entire vermilion zone, yet we can see some potential uniqueness in them. But they are not very easy to be tampered with. Means for fingerprints, somebody could have a false fingertip over it, or they could have hide their fingerprints. Like some people have Cheiloscopy, that is, they do not have fingertip like the furrows in their fingertips at all. Or if somebody is wearing gloves like me, so you cannot take out my fingerprints. Other than you are going inside these gloves to find out, which they would be smudged by now. So that is why lip prints give us an upper hand in this. So now coming back to our print, which is now dried. So I've already taken out my fluorescent powder, and now I'm going to dab this brush in this powder very lightly. Do not put too much powder on the brush, otherwise excess of powder on the print would also smudge it. So just put dab and do it like this. So the excess powder would go out. And similarly, when I'm putting this on the print, I'll do the same way. This has very soft bristles, so I can use it over my lip prints, but if you have a hard brush, do not put it directly on the prints. Try to use ostrich feather brush or any other animal feather brush here, because then you will be able to have that kind of softness by which you can. If you have a hard bristle brush, do not use it, because it will tamper your prints and you won't be able to see the prints at all. So avoid doing it a second time, because it is hard. If you will put another layer of powder over the same existing layer, then it might become hard to see later on. Now my prints are pretty much visible from here. You can also use a simple magnifying glass in order to see them. Now this was one part of the practical experiment, that was to develop the lip prints. Now I have developed the lip prints. Now coming on to the second part, that is to identify these lip prints. Now in 1960, Dr. Santos was the first scientist working on cheiloscopy who gave us a good view about classification of lip prints. So in order to know about what was the classification of these lip prints, so the first was straight line. That is, if you have straight lines, then they are considered to be type 1. Second is the type 2, that is curved lines. So if you have lines in C shape, they are called type 2 pattern, that is a curved line. If you have angled lines, that is they form a V or they have angular covers in the end or the beginning or at an intersection point, they are called angled lines. These are type 3. Coming on to the type 4 or the wave pattern. If you have anything related to a wave in your prints, that is the sign shaped pattern, that is type 4. This classification is no longer accepted. So till now this classification which we are using is classification of Suzuki and Tsuchihashi, which came in 1971, and this is till date use. Now this classification talks about the vermilion border to border lining. That means starting from this border line to the mid brim to the lower border line. So the type 1 is grooves. That is, we do not have any straight lines, like 90-degree lines are not exactly present in our lip prints. So they are talking about vertical grooves here, which are similar to straight line but not exactly a straight line. So the type 1 pattern is complete vertical grooves. A complete groove is one which starts from the border and ends at the mid brim with no breakage in between in the vermilion zone. That means it's a complete line. Similarly, even if the line starts from the mid brim and ends at the lower border, that is also a complete vertical groove. Now, type 1 dash, the next type is incomplete vertical grooves. Now, if a vertical groove is starting from the vermilion border and in midway stops without touching the mid brim, that is there are some breakage in between, then that is called an incomplete vertical groove. Supposingly a groove is starting from the mid brim but it ends before touching the lower border, that is an type 1 dash, that is incomplete vertical groove. The next groove is the type 2, that is Y-shaped or forked grooves. That means a groove which was going straight now gets bifurcated into two. So this Y formation in a groove states it to be type 2, that is forked groove. It can be on any side, upper lip, lower lip. It can be, the forked groove can be facing downwards, upwards, any form. If there is a fork formation, then that is a type 2 groove. Now, the type 3 groove, that is formation of intersecting grooves, X formation. This is the intersection point. Two grooves are coming and cancelling each other. Intersecting point is formed. These are intersecting grooves. Type 4, reticular grooves, forming a net-like structure or a mesh-like structure. That is, straight groove lines are going like this and they are being intersected by another batch of groove lines like this, forming a net-like structure. Type 4 is reticulate grooves. That means when the grooves form a mesh or a net-like structure, 3 grooves or more number of grooves coming vertically like this, being intersecting by horizontal grooves like this. This is a net or a mesh-like structure. This kind of a pattern, if observed, is type 4 pattern, that is reticular grooves. Now coming on to the type 5 or the undetermined grooves. That means other than the patterns we have discussed above, if there is another type of pattern which is unidentifiable by the table given by Suzuki and Tsuchihashi, then it is considered to be an undetermined pattern. Hence, it gets classified under type 5. So these were the classifications. Now, if I observe my pattern, there are incomplete vertical grooves in it, from both the mid-brim to the lower vermilion border, similarly from the mid-brim to the upper vermilion border. There is no other pattern formation, and some forked grooves can be observed on the upper vermilion border. Hence, this is a mixture of type 1 dash and type 2, that is forked grooves. So, the result is what we observed, that is type 1 dash pattern is seen in the lips. We took sample, that is my lips. So they have type 1 dash pattern. Now, as I told you the complete procedure, the theory of it, now I am going to tell you some tip points that you need to remember while you are doing this experiment. So, the precautions you need to take is, first and the most prime precaution that you need to take always while you are performing an experiment is your own protection. I couldn't wear the mask because I was taking my own samples, but you should always make sure that you wear a mask. Next is that while taking the samples, be very calm and patient. Do not take patterns very swiftly, pull or push the paper, because that could tamper the lip prints and also make the pattern smudge. Secondly, do not put the paper anywhere. In case the lip prints come in contact with any liquid or any other chemical, it might destroy or submerge the patterns. So make sure you are drying it on a dry palette or a dry plate. Then do not put it under sunlight directly or use any external heat source. Another is, in case your lip prints do not come, do not get self-worried that you don't have lip prints. At times, the lips are not in a complete state to give your patterns completely. Like I told you, they might be too dry or they might be overly moisturized. We do not have any sweat glands present. Any sweat pores are not present on our lips. So sweat is not a problem in lip prints, but at times excess of salivation results in incoming of pattern, that is, either the patterns are not properly seen or they are very much submerged to be identified. Also, I am using this fluorescent powder. So in case you do not get your patterns by fluorescent powder, you can also try using charcoal powder, or black fingerprint powder can also be used. Also, much of the percentage of good results can also be seen by using magnetic powder. So in case you have those powders available, you can definitely try taking lip prints from those powders. Now this is the end of the session. I hope this session was very much knowledgeable and you all will definitely try to perform this experiment, and hopefully you all will have good results. Stay tuned to our channel. Do subscribe, like and comment, and click the bell icon downstairs so you always get notified when we come alive with another session. So there will be a long list of practicals in this new year. I wish you all a very happy new year and stay tuned to AFRS. Thank you.